Hello, everyone. Thank you for inviting me to present at the annual NAPPC International Conference. My name is Juliana Rangel. I'm professor of apiculture in the Department of Entomology at Texas A&M University. And today I'll be talking about the nutritional ecology of honeybees in a changing landscape, particularly on work that I've been doing with my colleagues, former PhD student Dr. Pierre Lau, former postdoc Dr. Pierre Len, my current co-project director with our USDA NEPA funded project, Dr. Spence Beamer, who's also a professor of entomology here at Texas A&M University, our current postdoctoral fellow, Dr. Kanan Mani, and our PhD student, Mr. Mustafa Boshkuz. I also want to acknowledge our funding agencies, NAPPC and the Pollinator Partnership, for inviting me to present. Managed honeybees contribute an estimated $16 billion annually to the U.S. economy, primarily through crop pollination. Depending on the year, there are an estimated 2.6 to 2.8 million managed colonies of honeybees in the United States, over half of which are used for pollination services, particularly of almond in California in the months of February and March. Honeybees pollinate over 100 fruit, vegetable, seed, and nut crops, which are essential to the production of approximately 1/3 of the food that we consume on a daily basis. However, despite their importance, we have been seeing unprecedented losses of colonies year after year in the last few years. In particular, in 2024-2025, U.S. beekeepers reported a loss of over 55% of their colonies, which was the highest annual loss ever reported, far above the 14-year average of 41.4%, which is denoted on the graph at the bottom with the dotted red line. So as you can see, in the last couple of years, the average colony loss annual, which is in the shaded red region, has surpassed that already high 40% loss. These numbers are exceedingly high, in particular for some commercial beekeepers who reported from the middle of 2024 to the end of 2024 and beginning of 2025, losses of over 60 to 80 to even 100% of their operations. And they reported in particular issues with high Varroa mite levels. Varroa destructor is an ectoparasite, a mite that not only feeds on the fat body tissue and hemolymph of developing bees and adult bees, but also it's a very efficacious vector of many honeybee-associated viruses, including the formwing virus. So last year, several scientists went to the call of trying to understand what caused such high losses, and especially some scientists at the USDA Bee Lab in Beltsville, Maryland, who collected bees from collapsing and collapsed colonies, as well as hive products and parts to try to determine what was causing such high losses. They discovered that most of these colonies that had collapsed or were on the verge of collapsing had lost, had really high Varroa levels above the 3% threshold level, and had very high levels of honeybee-associated viruses, which are vectored by Varroa mites. And they also found that there's an exceedingly number of colonies that have mites that have become resistant to the most commonly used miticide right now, which is Amitraz. And so that is surprising and also troubling because it's one of the few miticides that tend to work at all times of the year, especially for commercial beekeeping operations. And so given that the bees are not, or the mites are not responding to this treatment, this can become a problem for the years to come until we find better solutions to the control of Varroa. As you can see from the abstract of that paper that said that viruses and vectors are tied to honeybee losses, other stressors such as nutritional stress and agrochemicals may have also played significant roles in these losses, and scientists are still working on trying to understand whether and how these factors are affecting high losses in the United States. So it's not surprising that colonies that are transported across the country are faced with several stressors, including, as I just mentioned, parasites like the varroa mites, other pests, low genetic diversity, the stress of transportation, environmental stressors, high pesticide loads, pathogens like viruses, but also bacteria and fungi, and in particular to this talk, poor nutrition, including monocrops, habitat loss, and suboptimal nutritional supplements. So for the topic of this talk, I'm going to focus on nutrition. So what are the building blocks of honeybee nutrition? As all living organisms, honeybees consume water, not only for their physiological processes, but also for the dilution of brood food and for processes such as evaporative cooling when temperatures are very high. They consume floral nectar as their source of carbohydrates, and they consume pollen. Pollen is the main source of several macronutrients, including protein, lipids, and digestible carbohydrates, as well as minerals, including macro and microminerals that are used in very small amounts, but indispensable amounts. Some of those macronutrients include phosphorus, sodium, and calcium. Some of the micronutrients include manganese, iron, copper, and zinc. But what pollen foragers go out and collect in the field, which is pollen, is not what actually nurses feed on. The pollen foragers bring back that pollen and then they pack it into open cells and create layers of this packed pollen that is now called bee bread. So through this packaging process, the germination of the pollen grain is prevented and the content of protein in the pollen is preserved. Not surprisingly, the nutritional content of bee bread is influenced by the landscape, the microbiome surrounding the landscapes and the hives, and the floral sources that are available at different times of the year. In our laboratories, we are working on a USDA-funded project that is looking at creating a multidimensional analysis of pollen collection, bee bread fermentation, lipid regulation, in particular fatty acids, to link a colony's nutritional input to colony performance using what is known as the geometric framework for nutrition. We have three objectives. The first one is to perform a nutritional analysis of bee-collected pollen by pollen foragers and characterize the nutritional content of pollens from different landscapes, including agriculture, urban, and rural. The foragers then collect the pollen and store it in the hive for fermentation, as I mentioned previously. And so our second objective is to look how in-hive pollen processing changes the nutritional content of bee bread, which is what nurses feed on to then turn into brood food that they give to the developing larvae. So our third objective is to see whether and how lipid intake regulation, in particular fatty acids, limits or augments colony performance in terms of colony growth and survivorship. But for the purpose of this talk, I'm going to focus on the first objective, which is to perform a comprehensive nutrient analysis of pollen across agricultural, urban, and rural landscapes to understand the honeybee's nutritional space. So we will have, by the end of next spring, 2 years of data collection from spring, summer, and fall, during which we will have collected pollen and bee bread from 5 experimental colonies per site to look for its protein content, lipid content, fatty acid content, and the content of 10 physiologically important elements. We have performed geographic information systems or GIS analysis to select our 5 agricultural sites throughout the United States, 5 urban sites, and 5 rural sites. In terms of our agricultural sites, we are collaborating with scientists at Oregon State University, the University of California, Davis, and Michigan State to collect meadow foam and cherry from Oregon, almond from California, watermelon and cotton pollen from Texas, and blueberry pollen from Michigan. In terms of the rural and urban sites, we are collaborating with volunteer beekeepers that live either surrounding the College Station and Bryan areas for our rural sites, or a beekeeper in Northwest Houston that owns and operates 5 apiaries within the city limits of the city of Houston. So as I said, we have 2 years of data, including those for 2024 and 2025 for spring, summer, and fall. And we are doing colony assessments to determine the growth and survivorship of colonies. So we're collecting an estimate of the worker population in each colony at each time point, the amount of brood present in the colony, the amount of pollen stored, and the amount of honey stored. But we're also collecting other information. So I don't have time today to give you the results from our colony growth assessments, but just so you know, we are assessing every frame in a colony, and each side of the frame is divided into 10 segments to then take the percent coverage for each of those metrics measured. So for example, we have one side of this frame divided into 10 quadrants, and we can estimate that this frame had 40% capped brood, 20% open brood, 10% honey, 20% pollen, and then we do the estimation of the number of workers present in each frame. We also collect very important data and samples. So we collect the corbicular pollen from pollen foragers by using front porch pollen traps. Then we use the same colonies 1 week later to collect bee bread because it takes about 1 week for pollen to ferment into bee bread within a frame. And then we collect adult workers from each of those hives to then have some metrics of health and gene expression analyses that we will be doing later on. But for the purpose of this talk, in terms of nutrition, we have taken the corbicular pollen as well as the bee bread to conduct nutrient analysis using several methods to quantify the amount of protein, lipid, carbohydrate, and elements within those samples. So I'm going to give you some of the results. I'm going to start with those in the agricultural pollens. Surprisingly, even though I told you that we had collected pollen from cotton and blueberry fields, when we actually did the taxonomic identification of the pollen, we discovered that none of that pollen actually came from cotton fields or from blueberry fields, but actually from plants that are surrounding the fields, which is interesting in and of itself. In terms of protein content was highest in almond pollen and lowest in watermelon pollen. So you can see here almond pollen, and it was a very tight spread. So all of these samples were collected in triplicates, and then a little wider spread in watermelon pollen with about 16% on average. And we found significant difference between most types of pollens in terms of protein content. When it comes to lipid content was higher in watermelon pollen with about 33% average, and almond pollen with an average of about 25%. Total lipid ranged from about 5% to 33%, often even for the same crop. So you can see the spread in meadow foam pollen. No differences were found between most pollen types in terms of the lipid content, however. In terms of carbohydrate, total carbohydrate content in pollen ranged from about 40% to 80%, often again for the same crop, particularly watermelon and cherry. But we did not find any significant differences in the amount of carbohydrate between pollen types in agricultural pollens. Now I'm going to move on to our rural and urban pollens, and we'll begin with protein content. So as you can see from these violent plots, we have on the left the urban sites and on the right the rural sites. We have data so far only for summer and fall. And we discovered that protein content was higher in the summer compared to the fall, and total protein amount ranged from about 10% to 27%, with no differences in protein content being based on the type of site, in being urban or rural. In terms of lipid content, we found that lipid content was higher in the fall versus the summer. The lipid amount ranged from about 19% to 43%, so it was a wide range in lipid content, and it was higher in the rural areas versus the urban sites. Finally, in terms of carbohydrate content, we found that carbohydrate was higher in the fall, with total carbohydrate amounts ranging from about 25% to 80%, with the seasonal differences being greater in rural landscapes compared to our urban sites. So for the corbicular pollen in urban and rural sites, we found that protein was higher in the summer, lipid was generally higher in the fall and in rural landscapes, carbohydrate was higher in the fall, and bigger seasonal differences were seen in the rural landscapes. And then we found that data that I don't have time to show you today, that macro and micronutrient content was not significantly different between seasons or landscapes so far. So we are in the process of performing DNA metabarcoding for all pollen and bee bread samples from across the seasons. So we extract the DNA from the pollen and the bee bread, we clean it up, we measure its amount, quantity, and quality, and then we send it off to the Texas A&M University's Genomic Core facility so that they can send us the sequences of all the taxonomic groups that these pollens belong to. Here are some interesting preliminary data on the most highly occurring plant families in the pollens that were collected both from urban sites and rural sites in both the summer and the fall. So in the summer, we found that in both urban and rural landscapes, there was a lot of Lathraceae pollen, including about 32 genera with more than 620 species of herbs, shrubs, and trees, including crepe myrtle, which is a very important aesthetically pleasing urban tree that is used in Texas in the summer. We found that also in the summer, Poaceae is a very important pollen generating family for honeybees. It's the family of grasses that includes wheat, rice, maize, barley, and sugarcane. And we found that it is also widely used by bees in the summer, both in the urban and rural landscapes. And conversely, in the fall, we found that the overwhelming majority of the pollen collected in both urban and rural sites belongs to the Asteraceae family, which has over 32 species of herbs, shrubs, and trees, including sunflowers, daisies, dandelions, and chrysanthemums. So some of the take-home message for our studies is that there is significant variation in the type and amount of macronutrients and minerals in corbicular pollen based on landscape type and seasons, which is not surprising because different plants bloom at different times of the year and different locations. This argues for a more precision nutrition strategy that we should undertake to enhance honeybee health and pollination services across landscapes. Our other type of work that we're doing in terms of trying to determine whether bees can discern the quality of their food in terms of the pollen that they collect, foragers collect in the field, is telling us that bees actually can make concerted decisions as to what type of protein and lipid content they prefer in their diet. And so this calls for a better creation of better pollen supplements or substitutes, because in times of dearth, these substitutes provide minerals, proteins, and other nutrients to honeybees that help them increase colony growth, particularly by allowing nurse bees to feed on these products that then lead to the development of a higher or bigger hypopharyngeal glands to produce high quality brood food. When you go to the market, you see that products have really high, these supplements have really high crude amounts of protein, but not very high amounts of fat or other contents. And this kind of is going against all of the findings that the amount of lipid and the type of fatty acids within the lipid fractions is higher in bee-collected pollen. So we need better supplements that better mimic the bee's macronutrient needs and preferences across seasons and various landscapes. So again, we are continuing this work to look at pollen collection and bee bread fermentation, and how the nutritional content of bee collected pollen and bee bread fermentation leads to better colony growth. It is not surprising, and I'm speaking to the choir, preaching to the choir, that honeybees require a plethora of multifloral pollen available all times of the year. But when this is not possible and we have pollen dearth, we have to be better equipped at feeding our bees with the necessary nutrients so that they have better colony performance and higher survivorship. So I want to thank you for your attention, to all of our funding agencies and our collaborators for their work and funding. And we welcome you to join our Facebook page, Tamu Honeybee Lab, which is our source of information about all the work that we do at Texas A&M University regarding nutrition and other bee-related topics. Again, if you have any questions, please send me an email. And otherwise, thank you so much for your time and attention.